Plasmids, and we talked about how two RNAs regulated plasmid copy number. It wasn't. It was a different concept, if you like, than just think about the way that nature can do these things, right? So for the Lac operon, we had a Lac operator, right? And this was essentially a binding site that had two parts that a dimer could bind to, right? And we had cooperative binding, and then we had a loop. So that's one way that nature can have a repression, right? You got a protein binding to a DNA site. Another way is you can have an RNA bind to another RNA. And there's a lot of people that believe that the initial world was an RNA world, although it's by no means certain, because we have no idea really. But it seems that RNA could have been the initial life form, for example, not DNA. And one reason to believe that is obviously ribosomes. Protein synthesis is catalyzed by RNAs. And so if you think about RNA, and what we just showed is that we had a piece of RNA 1 and RNA 2 that can interact, especially via these, actually there are 3 bases, just like codons. These guys have structure and they tend to interact here initially, and then that can cause a complete annealing, because remember, if you think about it, RNA 1 and 2, 1 here, 2 here. 1's the inhibitory RNA, 2 is the RNA that we're going to cleave with RNase H to initiate replication. These guys are being transcribed in opposite directions, therefore these two RNAs are complementary. You know that, right? So one is really anti-sense to the other and complementary. And that RNA-RNA interaction, we're going to find in this lecture just in a moment, governs other kinds of interactions. And the other one that I don't have time to talk much about in class, but you should be aware of this, is let's say we have some gene X, and as you know, there's a ribosome binding site, RBS. So when the RNA is made, mRNA, that codes for protein X, there is a ribosome binding site on the RNA. And what does that ribosome binding site do? Well, wow, let's see, the ribosome binding site in the mRNA binds to another RNA. What does it bind to? 16S ribosome RNA. You guys remember that? I mean, it's not magic. The 16S ribosome RNA binds to the ribosome binding site. The ribosome binding site in E. coli has something like this. It doesn't have to be AGGA, but that a mix of heavy G's and A's that can actually have complementary base pairing to the 16S ribosome RNA. So here we have another RNA pairing, right? And then it turns out that in the cell, for many genes we're recognizing, there are small RNAs that are non-coding. And when I say small, I mean less than 100 nucleotides usually. And I don't have time to talk about these, but I think I've mentioned them. Small regulatory RNAs that can bind to the ribosome binding site because they're complementary. And what do you think they do? They can block translation, right? They can block translation. In fact, these small RNAs are apparently, we don't have enough data to really say this definitively, but I'm pretty sure it's true. They are hugely important in human gene regulation and bacterial regulation and the regulation of every single free-living organism on Earth. Small RNAs. Now, that used to be known as junk DNA. Codes for a huge number of small RNAs. In E. coli, they found over 100 small RNAs that regulate gene expression. And they can regulate gene expression by binding to a ribosome binding site. And the reason I'm mentioning all this is because I want to talk a little bit more about RNAs and how they regulate, they're involved in plasmid-conferred regulation. Just a moment. But if you recall, we were talking about plasmids, right? And I think the last part, I talked about how RNA1 and RNA2 regulate the initiation of replication. Because there's an RNA primer, right? RNA2 gets cleaved by RNase H, but the RNA2 can be bound by RNA1, which inhibits its activity. Okay, so that's we're going to come back to that. So I'm going to ask you some questions about that and see if you have been thinking about this. So it's coming. So there's a few problems you have if you're a plasmid. Now think about this. If you're a plasmid, let's say you've got 4 copies of the plasmid, right? And this is your bacterial host. Now we're thinking like we're a plasmid. Sometimes it's useful to put yourself in the place of the plasmid. In fact, there was Richard Dawkins, this is a little bit before your time, you probably heard about selfish DNA. He came up with a book titled Selfish DNA, the idea that all we are is we're vessels for selfish DNAs, which are using us to carry them around and pass them from generation to generation. You could also think of us as vessels. All we do is carry bacteria around and pass them around and carry them and make a nice environment for them to grow in. And they actually control us. Crazy thought, but actually there's some evidence that parasites can actually control your behavior. And you get into parasitology, it's pretty amazing. So I mean, that's not such a crazy way of looking at the world, even though we're very anthropocentric. We think the world really revolves around us. But maybe not completely. But here we are, plasmids. Let's think that we're a plasmid, right? We know our host is going to divide. Okay, so as you know, the bacterial chromosome has to replicate, so there's 2 chromosomes just before replication, and then we have binary fission, and you get 1 into each cell. And actually, obviously, do something like this, and binary fission. And you got a chromosome in each cell. Fine. But what if you're a plasmid? Oh, man, let's see. It's going to be a little bit tough here, because if we're a 4-copy plasmid, you know why there's 4 copies now, right? Because when there's 1 copy, there's not enough RNA1 to repress. It gets to 2, it's building up. 3, we're slowing down. 4, we have enough, so RNA1 is binding to RNA2 so completely we have no more initiation. Does everyone get that? It's a pretty straightforward repressor hypothesis, and it turns out to be true, and here's what we're going to do. We're going to expand. The cell's getting bigger, so if you like. And because the volume's getting bigger, we can actually expand to 8. Does that make sense? The cell's, you know how E. coli is going to scale, it'll be like this, and it's going to be something like this, and then binary fission, right? So as it expands, we're going to have 8 plasmids. They'll be separated, they're going to be spread out through the cell. And what we want to do is we want to put 4 plasmids in each of these, right? And so we got to have some way of putting plasmids, knowing which plasmids kind of going into which cell, because what if we did this? What if we made a mistake and we don't know the difference between the 2 daughters and we put all 8 in there? This cell, we say it's cured. If it's a phage or it's a plasmid, we can say it's cured, basically cured of the plasmid, kind of like the plasmid was a disease and we cured it. That's the idea. So we got rid of it. We have no plasmid in there, right? Now, would this be a good strategy if you're a plasmid? Really, because then you're going to have cells that don't have you anymore, and this is a problem. And you're going to see some strategies coming up for how you make sure that the next daughter, all the daughters have you. But one strategy is to have a partitioning system, and I'm going to get into that in just a moment. But first I want you to tell me the probability of having curing. What if I asked you, what's the probability of 1 daughter being cured? What's the probability? Well, it's not too bad. For the first plasmid, what's the chance, let's pick this daughter. What's the chance that that daughter is going to get the first plasmid? 1. What about for the second plasmid? Right? So you kind of know what the answer is going to be. So what would the probability of just the one of these daughters curing? No one's saying anything. 1 out of 256. 1 out of 2 to the 8th. Does that make sense? What's the chance that some daughter is going to be cured? I said the chance of 1 daughter. Come on. The chance that either one, that at least one of those daughters is going to be cured. 1 out of 1/2 to the 8th divided by, or sorry, multiplied by 2, correct? Because you got one daughter and the other. They're both possibilities. If that's hard for you, all you got to do is go back and try this problem with a copy number of 2 and then work it out for yourself. Copy number 2 and just work out all the possibilities of where you could put those 2 plasmids, and it's really straightforward. So that would be 1 out of 256 for any given daughter and 1 out of 128 for a curing probability. Okay. If you have a plasmid and you don't know anything about it, and you carry out an experiment to determine if there's curing or not, if you see some probability around this probability for a plasmid, let's say they had 4 copies, then you would know that that plasmid doesn't have any partitioning system. And that's how partitioning systems were discovered. And we call them PAR. Some plasmids, especially low copy plasmids, in fact almost all low copy plasmids, have partitioning systems. Generally, we don't even know all the real details, but generally what they do is they allow a plasmid to actually bind to a membrane. So if this is our cell, and here's a plasmid, and actually the partitioning system allows it to interact with the membrane, which is involved in partitioning to make sure that it's going to bind to a membrane position either in one of the daughters before cleavage. And the partitioning systems aren't perfect, but they allow the plasmid to kind of know which daughter is which. Now, exactly how that works for any given plasmid isn't totally clear, but it definitely involves binding to the membrane. And we call them PAR systems, and they're very useful for low copy plasmids, because if you're low copy, like 4 copies, we call low copy. So what do we mean by low copy? Low copy would be like F plasmid we talked about has a copy of 1. PFC 101, you don't need to memorize this, but if you ever get into my field, that you will know this is about 4 copies. That's low copy. That's low. High copy is anything above 50 or something. PBR 322 we talked about has about 50 copies, 40 to 50 copies. That's sort of medium copy. High copy, 100 copies, 200 copies. Mostly, if you go to a laboratory anywhere in the world, and especially if they're studying eukaryotic gene expression, they'll use high copy vectors because they want to be able to purify DNA easily, and a high copy vector gives you a lot of DNA, plasmids, or whatever you're purifying. You don't want low copy because you don't get a lot of DNA. But there's disadvantages to high copy plasmids. Gene expression really screwed up in a high copy plasmid. So if you're studying gene expression of your bacterium, you don't want high copy plasmids. But if you're a eukaryotic or investigator, you don't care. Use bacteria as a tool, who cares? Make a lot of plasmid. All right, so that's partitioning systems. And I think if this doesn't quite make sense to you, as I said, go back and try copy number 2. See how that works. Okay. There's another way to deal with this. And I like this way. I think it's really cool that nature developed this. Partitioning is great. Okay? Partitioning. F plasmid. We're going to talk more about F, as I said. I've mentioned it. The fertility factor. It's a copy number 1, and it has a partitioning system. But in addition to that, it has what I would say, if I'm thinking of as a plasmid, an evil system. A really nasty, evil sort of, it's like, what was that? There's a movie with Glenn Close. What was that called? Fatal Attraction. Yeah. So if you can't have your lover, you kill your lover. If you can't, if one of these cells, one of these daughters loses you, you just kill them. Just kill them. I mean, what better way to ensure that every one of those daughters is going to have you? If they just happen to not have you, they're dead. Sort of vindictive, right? So, of course, that's very, very, anthropomorphizing, if you can say that word, but it's thinking about a human sort of thing. But it really is a very interesting system. And another way we can call these, they've been called addiction systems. And I'll give you one that I always give, but there's many, many. So basically, another way is I call this kill a cell if it dumps you. That's kill a cell if it dumps you strategy. So here's what we're going to talk about. This particular one is Hawk and Sock. Hawk and Sock. Hawk is host killing. So we always have, 3 letters for gene. This would be the proteins. Host killing, Hawk. And Sock is suppression of host killing. So this is sort of, think about this. This is the toxin. This is the antidote. There are many, many systems like this. They all work in the same strategy, but they use different kinds of ways to kill. So here's what's going to happen. And I'll just give you an overview, and then we'll show how it works. Here's the idea. You're a plasmid. And what you do is you make a nasty toxin all the time. But the host that carries you isn't going to die because you're making an antidote. So it's like, the host doesn't know. You've got a toxin and an antidote. The host, which is your bacterial strain you're growing in, grows fine because you've got a toxin, antitoxin. They negate each other. Now, let's say you've got a daughter cell division. You've got 2 daughters. And what happens is you lose this plasmid in one of the daughters. The thing is, both the toxin and the antidote are being encoded by genes, and they can either be RNAs or they can be proteins or whatever the toxin is. The toxin tends to be pretty stable. The antitoxin tends to be very unstable. So if you constantly make the antitoxin, you're in good shape. But as soon as I'm a daughter and I have no plasmid, I've got some toxin DNA, or some toxin protein, in me, and I've got some antitoxin. But I have no plasmid to keep making either one of them. So what happens is you start seeing the antitoxin, the antidote, start diminishing because it's unstable. And then the toxin starts becoming active, and I croak. I'm dead. And the plasmid, from the plasmid standpoint point of view, this is fantastic. Because then, in the population, it's going to be retained. Because I know my line is dead. The line that has no plasmid is dead. So the only lines of daughters left are the ones that have plasmids. So how does it work? For this guy, if this is the DNA, so for this what we've got is Hawk would be here. So we have just, as you would expect, let me separate this more so you can see what's happening here. These are the 2 strands of DNA. We always do. And then we make an mRNA, which is going to be coding for this toxin, Hawk. So this, 5', 3', mRNA. Now, same kind of idea, what we talked about with regulation of initiation of plasmid replication. We have a antisense, or opposite, RNA, made in the opposite direction, and this is Sock. So Sock is an RNA that doesn't code for anything. But what Sock is going to do is it's going to bind to Hawk, and it's going to block its translation. And the way that works, again, the idea is that you've got these domains of the 2 that interact because RNA has structure, and these, I call this kissing, where they come together. That is also happening here. So again, we have 2 RNAs that are regulating something. And so what happens is Sock will block translation, in this case, of the toxins. We don't even have an active, so it's not like you make a toxin and then an antitoxin binds to it and inactivates it. We don't even translate this toxin unless Sock is gone, and Sock is unstable. So Sock RNA is unstable, and the Hawk mRNA is not, I'm not going to call it stable. There aren't any really RNAs in bacteria that are that stable, but it's more stable. Let me clarify that, because you may not know this. I've never talked about this, but The RNAs in bacteria tend to have half-lives on the order of minutes. That means half of the RNA is gone within 5, 6, 7, 8 minutes. Eukaryotic cells, like our cells, RNAs have much longer half-lives. Okay, half-lives in hours. Generally, more stability. Okay. So in some ways, you can think of this as like a little time bomb where if this Hoc RNA doesn't have any antisense sock to block its translation, you're done. You're done. You're going to translate. And I think that many of these toxins put holes in the membrane so they work kind of like some phage work. If you're a phage and you replicate like T4, have you thought about this? How T4 replicates, maybe you've got 100 phage in a cell. It's got to get out, doesn't it? And bacteria are pretty strong. They've got a cell wall that's really tough. And so what they have to do is poke holes, usually holins. They call them holins. Why not? Holins. They poke holes in the cell. And they cause leakage out, and they cause the cell to blow up because the cell is leaking all of its contents. So that's how most of these toxins and phage work. Yeah?
So if the Hoc gene isn't getting into the daughter because the plasmid's not getting into the daughter, then why does it not?
Well, because you've got Hoc RNA.
Oh,
Hoc messenger RNA is there, right?
Oh.
And so a messenger RNA split equally between the daughters. So the two daughters have Hoc messenger RNA, plus they have Soc RNA. But the Soc RNA degrades much more rapidly. So within a few minutes, Soc RNA is gone. Right? Right. Okay, now I'm going to present a concept for you. It's called incompatibility. Another one that you could make, I'd like to make the human analogies only because one of my ex-professors, Stan Falco, when I was at Stanford, was fantastic because he'd teach medical students and he'd have them memorize things by having some crazy thoughts attached to the different, if you ever teach, this is kind of, I think, a useful way, make people think about something that they can relate to, and then you kind of categorize it. So incompatibility between people, obviously, if two people that are exactly alike may drive each other crazy, right? Because they both have the same faults and they recognize those faults. Two people that are maybe opposite, they tend to attract. Opposites attract, that's a plus and minus. That's true for sort of electronegativity. And same kind of thing happens here. So we call this incompatibility. What this means is we say that if two kinds of plasmids, use the same copy number control mechanism, and I mean the same, they are, what do you think? Compatible or incompatible?
Incompatible.
Yep. Incompatible. I spelled it right there. They are incompatible. They're incompatible. Okay? If they're different systems, they're compatible. And I'm going to give you a little problem in a second to make you really think about what that means. So I'm going to give you a hypothetical here. Do you remember back? So back, we just talked about it with choline-1, and it has RNA-1 and RNA-2, and this RNA-1 binds to RNA-2 and inhibits, it basically pulls it away from the DNA and doesn't allow it to have an RNase-H cleavage, and we can't replicate. So it's a repressor, right? What if I took that region, let's go back, and we had a region, and I know you remember this, where RNA-2 was made. Same as, basically, very similar to this, right? It's the same drawing. So, RNA-2, RNA-1. What if I just took a section of this where the kissing occurred, especially, that would be really part of it was critical. I took and just changed some regions here, just even one major region that is required for them to come together, and I just changed the bases to some other, I just took a region of 10 base pairs, let's say. You can even do this with one. But 10 base pairs, and just replace this with some other 10 bases right here. Right where the two come together, I take and I just, I may replace the bases. It might have been a G, G, C, A, C, T, A. That might have been a key region. Now this is at the DNA level, and of course you're going to have transcription. RNA2 will come in, and RNA2 will basically be this, won't it? But you'll have a U here instead of that T. You guys agree? What if I just took this region and I made it C, A, G, T, T, A, C? What if I just popped this DNA in? I can do that with genetic engineering. Talk a little bit more about that. What if I just took this and replaced it right for that region right there? That's all I do. That's all I do. And I don't even know what base, I just random, we could do this experiment. So if I did that, what is that going to do? You think it's going to really screw up the copy number? Is it going to change the incompatibility? Will a plasmid that has our new region be compatible or incompatible with the original one that we started with? These are interesting questions, right? Start with the first one, second one. Is it incompatible or compatible? Will they use the same machinery? No, not really, huh? Because the RNA1 is different and the RNA2 is different. So what that means is that this new RNA1 that's from our new plasmid that has this sequence will not hybridize, will it, to the old RNA2. So I could say the RNA2 parent, or let's say old. It's not really a parent. Let's say the old. And the RNA2 and the RNA1 new, that's our new one, the RNA1 new will not bind to the RNA2 old. Does that make sense? Because we changed the base pairs, didn't we? But the new RNA2 will bind to the new RNA1, won't it? It has to, because we use the same, we put in this new duplex, and therefore, this would be, this will be RNA2, and this will be, and the RNA1 sequence will be basically that, right? And they'll hybridize, won't they? So we just created a new compatibility group, didn't we? Did we? And what does it mean, compatibility group? What do I mean by that? And you can keep thinking about that, because I'm coming back to it. If I have two plasmids that use exactly the same machinery, now let's say we have two plasmids, they have the same machinery. One I'll put by just this color and one blue. And one has, let's say, tetracycline resistance. I put tetracycline on it. The other I put chloramphenicol. Remember that one that you all said, what the heck is that? So they have different antibiotic resistances, right? If I just grow this bug out, and it makes lots of daughters, and I grow it overnight in liquid broth, and I put only tetracycline as selection for this guy. What incompatible means is after a few generations, what's going to happen is the only plasmids you're going to find in the daughters are the ones that had the tetracycline, because the ones that had chloramphenicol, which we can easily find out if they're there, right? Because we can just put them on chloramphenicol plates to see if the plasmid is there. Does that make sense? So what's going to happen after a while is they use the same machinery, and what's going to happen is this poor plasmid, the chloramphenicol one, is using the same machinery as this, but we're selecting for this guy, aren't we? And what's going to happen is this, all the RNA1 from this guy is going to end up inhibiting this guy eventually, because we're always selecting for this guy to be there. Does that make sense? Mm-hmm. But if we have this altered plasmid, and we label it, so this is, I call this new, and this is old, and the new one, we put chloramphenicol on it. What will happen if I select for tetracycline? Will I totally exclude the new one? I won't, because the RNA1 from this plasmid doesn't bind to the RNA2 of this one. Right? So as long as this new plasmid has some kind of a decent partitioning system, and there's some other things we'll talk about, it won't necessarily be excluded, so we'll have both the old and the new, even though we're only selecting for the old one. Does everybody get that? Incompatibility. So the reason it's important in the laboratory is if you want to have two different plasmids, one that contains some genes and one that contains another, what you want to do is make sure that they're compatible, which means they have to have different systems of replication. And in this case, we just changed a few bases and made a whole new incompatibility group. Do you get that? If you don't, the best thing to do is just go do this to this region and make it RNA1 and a 2 and realize that now we have new hybridization going on. Okay? Hopefully that makes sense. Okay. So a couple other concepts I want to give you. Let me just talk about a few things. I talked about, just in terms of plasmids, we're using these kinds of strategies to develop plasmids. And in the laboratory, you might see things like PBR322 was really one of the first plasmids. I said it's about 50 copies. You don't need to memorize this, but I want you to be familiar with these in case you see them in the laboratory. This uses CoLE1 origin. CoLE1 is the RNA1, RNA2. So everything that we just talked about is totally relevant for these guys. PUC, after University of California, they're developed at I think UC Davis. PUC is based on PBR, but they have hundreds of copies, high copy, and they are CoLE1, and actually you'll see them called PMB9, and they are mutated derivatives. And give me one mutation that could increase copy number, knowing what you know about CoLE1 replication, RNA1, RNA2, and a protein that has to be there to stabilize them.
You don't have the protein that stabilizes?
Yeah, and in fact, you probably want to have the protein, but let's make its affinity a little less. Let's make maybe it's the number of copies in a cell less, because if we don't have it, then we have like 1,000 copies per cell, and our cells like die. So we probably don't want that. But yeah, exactly. So we could have mutations in the ROP gene, and they're going to increase copy. And I'm going to go back to the question I asked you guys. I said I'd take the second part of the question. Remember, the first part of the question is, are you going to change copy number by putting this here? It's a little harder question. Are you going to change copy number by putting this here? I'll make it easier. And you'll know what I'm saying. What if I put this sequence in? And the original sequence was this. It's AT-rich. You know AT-rich melts quicker. You guys told me that a long time ago when we did DNA melting curve. Remember that? So the affinity is going to be less, isn't it, for the two RNAs to come together, don't you think? Because the affinity is based on hydrogen bonding, and you get 3 hydrogen bonds for GC and 2 for AT. So maybe what would happen? If the RNA1 affinity is less for the new system, what's that going to do to copy number?
Increase.
Yeah. So I would predict that this new region, that new region is going to have a higher copy. There you go. So if you want to change copy number here, you could change the base pairs. Because here the affinity is based on hybridization between the two DNAs, which is based on what? AT-GC. You get more energy, right, from a GC. So generally for plasmids, that's what the PUC plasmids have, those kind of things. And then F I talked about, that's 1 copy, and that's the fertility factor we'll talk about. And that's all I'm really going to mention, because you're never going to remember these other things anyway. But I want you to have the concept behind strategies so that you can actually do this. There's one other thing I need to talk about before we move on to recombination. And I want to just talk a little bit more about low copy and high copy plasmids. And you'll notice that on GATRO space I put some questions up. on plasmids, and I'll just talk about those, a few of those questions. The questions on gaucho space, I want you to look at that. I want you to think about it, and I'll probably just cover the first question. I want you to just look at the question and answers. That comes from Stan Malloy, who's a colleague of mine, down at San Diego State University, who posted those. Stan wrote a really good book on genetics, but hasn't updated it, so I don't use it for the class anymore. He used to be the president of the American Society for Microbiology. He's a really good microbiologist. And he's got some good questions about plasmids. I want to mention one more thing. Low copy versus high copy plasmids. And there's one more thing here, and it's going to segue into recombination nicely. So we got another problem, especially for, well, actually for any kind of plasmids. But let me just give you an idea. Low copy plasmids, generally I said, have efficient partitioning systems. So these guys have to have partitioning systems because they don't have the game of numbers on their side. There's so few copies that they, I just told you what the frequency with 4 copies would be. What about 1 copy? What would the frequency of loss be? And it's actually 1 out of 2 total if you think about either daughter. You can work that out for yourself. 1 out of 2. 1 out of 2 times you're going to cure. You'll lose it unless you have a Hock-Sock type system to kill all the daughters that don't have it. But the other thing that we find is that, well, let's over here. High copy, in many cases, we see no par system. And the reason is, imagine, and you can do this, you just go get a calculator. What if we have 20 copies? 20 copies. What's the frequency of curing with 20 copies if we have no partitioning system? 20 copies would go to 40, right? And therefore the chance of curing would be 2 to the 40th. That's a big number. Divided times 2. For the curing for either daughter. You with me? I don't know. It's Friday. 2 to the 40th. Now, do you think plasmid doesn't care? 1 out of 2 to the 40th times 2. I mean, come on. So that they don't even worry about partitioning systems. But there is something that both of these plasmids have to worry about. And here it is. And I don't think you probably thought about this, and I probably wouldn't have either. But this is what happens. Here's a cell. These 2 plasmids are homologous. They're identical. And as we're going to find out in this lecture, barely, and in Monday, is that the Rec A, B, C, D system, which is the same system that way back when it seemed like, when we were doing this, and we had recombination, that's all carried out by the system we're going to talk about. You know what it does? It recombines 2 identical plasmids together to make a dimer. And it can take the dimers and make them into tetramers as well. It can't go on forever because it turns out cells that carry like bigger than a tetramer, they don't grow very fast for various reasons. But the bottom line is you get these dimers. Now, what effect is dimerization going to have on curing? Let's look at a dimer. The dimer has 2 copy number control systems, the RNA1, RNA2 region. You guys agree? Right? So here's a question for you. I know I'm asking a lot of questions for Friday, but I want an answer to this one. Here we go. Go back to the original one that I talked about. I think it was here. I hope I still have it. Yeah, here we go. All right. So if I have 4 copies of a plasmid, right? If I have 4 of these, what if instead of that, they dimerize, and so I have dimers. So I'll have 2 dimers instead of 4 monomers. What is the copy? How many copies of the plasmid? How many plasmids will there be in that cell if I get 2? If I have 2 dimers, is the copy net will it be like this? I'll mark it like that. Am I going to have that? These are dimers, okay? It's a pathetic looking dimer, but those are dimers. Is that right? this copy number? Is it how many plasmids are there? How many pieces, actual plasmid DNAs? How many circles there are? What are we titrating? Do you know what I'm asking?
Is it RNA 1?
Do you know where you are? All right. So I said we had copy number 4. So in a regular cell, not in a 4 division, but just let's say there's 4 copies. Will there be 4 copies of the dimer? No. There will not. It will look like this. Why? There's only 2 plasmids. Because those 2 plasmids have 4 regulatory regions, don't they? And the regulatory regions, RNA1, RNA2, aren't those the guys that govern copy? Aren't those the guys that are being titrated? When I say the word titration, you know what I mean? I mean RNA2 binding to RNA1. RNA1 levels rise as we get increasing copy. Look what happens. Isn't this cell going to be making the same amount of RNA1 as that cell? Isn't that cell going to make the same amount of RNA1 as that cell? Because there's an RNA12 here, an RNA12 here, 1 here, and 1 here. There's 4. 4 nodes, if you like. 4 regions that make RNA1, correct? How many regions make RNA1 here? 4. Doesn't that mean the concentration of RNA1 will build up the same here as here if there's 2 plasmids here? Yes. And what is that going to do to partitioning? What's that going to do to curing rate? If we start dimerizing all our plasmids, our copy number, the actual copy number of the plasmids, right? The number of plasmids goes down. Therefore, you have an increase in the frequency. In this case, what increase in frequency would there be in curing? 2-fold? Wow. Okay, I want you to think about that one. And I want you to work on it. Think about it. What increase, if I go from 4 monomers to 2 dimers, what change in the curing frequency? These are the kind of questions that I know you won't actually solve that kind of question in your future life, whatever you do, but you have to be able to think like that. That's what you're gonna get a good job. They're gonna pay you for thinking about things like this. That's why I think they're important. So think about that. Yeah.
So what exactly is the process of curing?
Oh, what is the curing? Is just a randomization. So you got one daughter, or sorry, one mother, and the mother has a certain number. Let's say she's got 4 plasmids. So we have a certain. We're going to have to set when we put a septation here. If you like, we're essentially deciding: are we going to give a plasmid to this daughter, or are we going to give a plasmid to this daughter? So if we put all our plasmids, if that's how many we have, so this would be for a copy number 2, obviously, then it goes to 4. What chance are we going to have that we'll put all of them in one basket?
Oh.
That's all. It's a very straightforward statistical problem.
So wouldn't it be less? No.
I want you to think about this. You know what you do is you just take and do the probability for when these guys, remember, this would be the copy number, so this thing's going to divide and make 4. This one's going to make 8. We did the one for 8. I already solved it for you. You just have to solve this one. And then tell me what change in the frequency will result. And you're going to find out it's more than 2-fold. Okay? So try that. So, what you guys, see if you can think about this. And because we only have a little bit more time. This is a problem. Does everyone agree? If you start dimerizing, you're going to increase the frequency of curing, right? Now remember, we're thinking like a plasmid today. So we're thinking, that's not good, that's not good. So what do? I got homologous recombination bringing me together to form dimers. I don't want to do that. So I could inhibit the recombination system of the host, couldn't I? But that might not be too smart because then the host wouldn't be able to repair the damage and then I'm toast as a plasmid, right? I got to keep my host happy. I can't screw up my host. Screw up my host, then my host isn't competing well with some other host, and I lose. So here's what they do. Almost all high copy plasmids and even low copy plasmids have we'll just call them they're recombination sites. Let's see. I'll give you an actual one. what they've got is recombination sites for site-specific recombination. So let's just take a plasmid. Let's take, I think this is for chole-1 actually. I believe this is true. Yes, chole-1, the one we've been talking about, has a little site called SIR. And what SIR allows is it allows a recombinase, and you don't need to know this name, but this is a recombinase, and I'm going to talk about site-specific recombination maybe like in a minute if I get there. But this, what this recombinase does is this. These two guys came together, homologous recombination, right? They'll form one big plasmid. And I kind of drew it like this. So, you know, it's got two together. So it makes one big plasmid. Here's what SIR does. We've got a SIR site here. We've got a SIR site here. You agree there'll be two sites, one in each plasmid? This recombinase, which comes from the host, what it does is it can recognize this and catalyze recombination between the two. What does that mean? Catalyze recombination between the two. If we could look at the two sites, SIR on one and SIR on the other, what it does is it recognizes this enzyme, ZIRCD, recognizes these two, brings them together, and then it simply cuts this DNA and cuts this DNA, and it joins them like this. So it just catalyzes recombination. You know, we talked about the old recombination we talked about where two regions recombine. That's what it does. And it's site-specific because it only works at the SIR site. So every time a dimer is made, we just undimerize it. We make it into monomers by recombining between the two plasmids. It's pretty cool. And most plasmids have these sites. And if you don't have those sites in there, you're going to be in trouble. You're going to lose your plasmid. So as budding molecular biologists, if you're in some lab and they're losing plasmids all the time, just go find a SIR site, go look it up on the internet in PubMed, go find out what this SIR site is, make a copy of it, put it in your favorite plasmid, and you're going to have a plasmid that doesn't lose itself in the future daughters, a plasmid that's stable, a plasmid that doesn't start getting cured at a high rate. Because the SIR sites will make sure it doesn't form dimers. Okay. And I've only got a minute left, so is there anything? Let me just cover the very first question that's on the internet site, and that's all I'm going to do, and then I'll see you guys Monday. So here's the first question if you go look at that. Replication of plasmid chole-1, it's all about what we talked about, is catalyzed by the host-encoded DNA polymerase 1. I told you about that, right? In wild-type cells, POL1 is present in large excess over that needed for plasmid replication. Initiation of plasmid replication is downregulated by the plasmid-encoded ROP protein, we talked about that, which is present in limiting amounts. If the protein synthesis inhibitor chloramphenicol was added to sensitive cells that carry a chole-1 plasmid, what would happen to the copy number? We sort of talked about that, didn't we? And what was the answer to that? The answer, addition of chloramphenicol will prevent synthesis of new proteins. We said that. It's a protein synthesis inhibitor, including POL1 and ROP. But there's enough POL1 in the cell so that you can continue to initiate replication, but ROP is limiting. There's not much of it. So when ROP stops getting made, there's no ROP. No ROP, you don't have RNA1, RNA2 hybrids that are stable, in which case RNA2 is run away and you make thousands of copies. So if you want to make a lot of copies for a plasmid like chole-1, just simply add some chloramphenicol, wait overnight, you'll have thousands of plasmids per cell. I'll see you guys Monday. Oh, and then come up if you want your regrades.